Good afternoon, everyone. Please take your seats. Welcome to this Brace SIG sponsored session. So benefit risk and well, benefits and risks and in the regulatory context. My name is Marieke de Bruin from Utrecht University. I'll be opening this session. And then I will hand over to my co-chairs, Christine Holgreen from University of Copenhagen and Ken Bernwickel from Eli Lilly. So Christine, over to you to introduce the first speaker.
Yes, and thank you. And our first speaker is Hong Yang, dialist from FDA. So welcome.
Thank you. It's my pleasure to have this opportunity to present our benefit risk assessment in this meeting. I'm from FDA CBER, the Office of Biostatistics and Pharmacovigilance. This is my disclaimer. The presentation reflects the view of myself should not be constructed to represent FDA's view and policy. Also, I have no potential conflict of interest to disclose. FDA published a draft guidance in September 2021. In this guidance, described FDA's structured benefit risk framework for the new drug and biologic products. The framework includes 4 dimensions. The first 2 dimensions here are analysis of condition and current treatment option. These are considerations related to the therapeutic context, so it's disease specific. And then the later 2 dimensions is benefit and risk. And this is more product specific. So FDA, when we make a decision, we consider the totality of evidence, include this all 4 dimensions. We also consider uncertainty, consider how to make trade-off between the benefit and risk. And then we make decision based on if the benefit is outweigh the risk for the product. Historically, FDA has used real-world data and real-world evidence for the post-market safety monitoring. However, the real-world evidence and data has been used for support effectiveness on much more limited basis. In 2016, the 21st Century Cure Act was signed into the law to promote drug development to more quickly bring the innovation to the patient by leveraging the real-world data and real-world evidence. FDA developed a framework for use of real-world evidence in 2018, also published consideration for use of real-world data and real-world evidence in 2021. So the link to those guidance are all provided in this slide. Real-world evidence and real-world data and real-world evidence are crucial for benefit risk assessment of COVID-19 vaccines. First, the vaccine efficacy obtained from clinical trial may not directly measure the benefit of the vaccine in the real world. Because the benefit of the vaccine not just depend on vaccine efficacy, but also depend on the disease incidence among the population. And the vaccine efficacy demonstrated by the well-controlled clinical trial may not represent the vaccine effectiveness in the real-world setting. Also, a lot of time in clinical trial for the vaccine, we use the antibody titer as the surrogate endpoint. Therefore, the vaccine effectiveness need to be further confirmed in the real world setting. Vaccine effectiveness could change due to continued evolving of virus strain, and also the waning of the human immunity to protect after get vaccine to protect from the disease. And vaccine effectiveness may be different among the subpopulation, and some of them may be underrepresented in the clinical trial. And then finally, the real adverse effect, and also the long-term protection from the vaccine may not be appropriate assessed in the clinical trial due to small sample size and short follow-up. So in May 2021, Pfizer and BioNTech, they submit a biologic licensure application for COVID-19 mRNA COVID-19 vaccine for the indication of use among age 16 years and older. So FDA conduct benefit risk assessment to assist the evaluation of this vaccine. We follow FDA structure framework for benefit risk assessment. So for analysis of a condition, the COVID-19 pandemic, it posed a great challenge for public health. It caused, by that time, it caused 200 million cases of the COVID-19 cases and 4.3 million related deaths worldwide. Among them, 90% of the cases are occurred for the age 16 years plus. So at that time, when we reviewed this BLA, there is no licensed vaccine was approved. And there's no approved drug for treatment of COVID-19. And there is 3 vaccine is under emergency use authorization. So this is the Pfizer's BioNTech vaccine for 16 years older, Moderna vaccine for 18 years older, also Janssen vaccine for 18 years and older. And for the EUA, basically, it was authorization by FDA to distribute otherwise unapproved products under the public health emergency. So FDA grant these 3 EUA primarily based on the preliminary result from incomplete clinical trial that demonstrate potential benefit risk profile of the vaccine. And for the clinical trial, they demonstrate that the vaccine has a very high efficacy for this Pfizer's vaccine. It has 90% efficacy against the confirmed cases and 95% efficacy against the severe COVID-19 cases. However, the real world benefit of this vaccine is uncertain because I already mentioned earlier, the real world benefit of the vaccine also depend on the disease incidence among the population. Higher the disease incidence, and you have greater the benefit from the vaccine. But it is uncertain which direction of the pandemic will go. The incidence will become higher or going down. So there is uncertainty there. And also there is another challenge is at that time, there is a new emerging Delta variants. And so the clinical trial data is mainly based on the original variant strain. So there is unknown about the efficacy of the vaccine for the new emerging Delta variant. And also, we also observe among those vaccine recipients, their antibody titer are waning after the vaccination over time. And for the risk, the clinical trial did not report any notable serious adverse event and any deaths related to vaccine. However, both FDA and CDC identify elevated myocarditis and pericarditis cases rate among the recipient post-EUA. So the risk was identified higher, especially among the male adolescent. However, the data is very limited. So also some study data are inconsistent. And for risk management, FDA has option. Either you can provide risk information in the product label to communicate this risk to the health provider or the public. And also, FDA can enhance post-market safety surveillance to further monitor this risk. And also, we can work with sponsor to develop some research plan, study plan post-market. So a key regulatory question at that time for the BLA review FDA faced is, with this kind of the evidence, both benefit and risk, how do we consider if the benefit is outweigh the risk, especially for the high risk group that is male, young, around I think it's 16 years old to 35 years old. That is the major high risk population, especially for 16 to 17 years old. So because of this, our office was tasked to conduct a quantitative benefit risk assessment to assist CBER's review on this BLA. So our analysis focused on 16 to 17 years old male individual. We assessed the benefit for 1 million of this individual with 2 doses of Pfizer vaccine. And the benefit we consider here in this analysis include prevent the number of COVID cases. Okay. ICU stay and death. And the risk we consider any additional myocarditis cases related to the vaccination, related hospitalization and death. We structure 7 scenarios to examine different uncertainty, include the uncertainty on the disease incidence, uncertainty on the effectiveness of the vaccine, uncertainty about the rate of myocarditis cases. And here I only present this most likely scenario. So here is a summary of our assumptions. So if we look at here, our vaccine efficacy is coming from US International Epidemiology Study. And the incidence rate is coming from CDC's COVANet. Our vaccine attribute myocarditis case rate is based on data from CBER, BEST system and CDC's data link. And here is our result. So from here, you can see on the left-hand side is the benefit, right-hand side is the risk. You can see clearly benefit is always the risk. So based on totality of the evidence and our result, FDA make decision, the benefit is always the risk. The product was approved in November 2021. So with that, this is my knowledge slide. This is our team contribute to this benefit risk assessment also FDA review and CDC and the best system partner, they all contribute to this project. Thank you.
Is the one down there?
Thank you very much. I'm Ariel Arias, Université de Montréal. I'm former Health Canada employee as well. I retired there. But risk benefit has always been very, very difficult to handle. And in this particular context, with so much uncertainty around it, it's even more complex if you want. So thank you very much for the presentation. I'm just curious. One of the things that often I see missing, but I think it's missing. But anyway, I want to hear about you. Is the considerations around the uncertainty of the regulatory decision itself, not of the data or all the uncertainty about the effectiveness or the safety, but the decision itself. Being what would be the impact of having the wrong decision or not having the appropriate decision, let's say. I don't know if you get what I'm trying to say, but it's in the pre-marketing side, there is a lot of this when you are developing a new drug. And so it's now people are paying a lot of attention to that. So I don't know, what are you thinking about it?
Well, because when we make a decision, especially like this COVID-19, you need to make the action in timely efficient. You cannot wait until all the uncertainty is gone. So what we can do is based on the data available, considering uncertainty. Of course, each individual, when you're making decision, there is also a judgmental decision there. So we can say it's based on our data, try to make we think most reasonable decision at that time. So, yeah. Thanks.
Am I on the left? Oops. I don't know how to use the mouse effectively. Yes. Okay. Trying to see glasses on, glasses off. Okay, I think we'll stay on. Hi, everyone. I'm here to talk about a methodological question that comes up in a variety of epidemiology applications. This one is in the area of patient preferences and the sample. This was part of the IMI Prefer project. And I'm going to talk about a preference survey we administered, and we compared first-degree relatives with the general population. Here's my disclosures. I'm going to give you a little bit of background before jumping into the study. This study looked at rheumatoid arthritis, and preference studies themselves often use panel data of the general public. And if we can't find the right sample sometimes, or if we're trying to be efficient, we might pick a general public and say, assume you have this disease and assume that the drugs to treat might have a certain profile. And so preference studies often use panels, but panels might not be a good proxy for patients. So the other thing I want you to know about this example is a little bit about RA. There are a lot of studies that are actually starting to read out now with mixed results, but there was some evidence out there that early treatment for people who are at risk of RA could potentially delay or halt progression or diagnosis of RA. And one of the groups that it's at greatest risk are first-degree relatives. There are additional risk models out there, and you can be either 6% higher up to about 40% higher, depending on if you have biomarkers, symptoms that are sort of early RA sort of markers. So first-degree relatives have an increased risk of RA, but the general public is also at risk. So the objective of this study was to compare preferences of RA preventive treatments between the general public, which we recruited from the panel, and first-degree relatives. A little grounding about what is preference. Patient preferences simply measure what matters to patients. And there are ways to do this where you can do qualitative interviews or quantitative, and there are a series of choices that you can do. Simply what matters can be straightforward. If you want to know how much it matters and getting into ranking and weighting, it can be a little bit more complex. And then you can get into trade-offs that come out of models. And these, again, there's a whole bunch of different options here, but I wanted to just ground you on what do I mean when I say patient preferences for a treatment. There's a lot on this slide, but what I wanted to convey is we used best practices in designing preference studies. They're rigorously designed with regard to the sample. The key here is we had a general public sample and first-degree relatives that we recruited during the pandemic based on their family member coming to an RA clinic or participating in an RA study. The study design is fairly standard. It was a discrete choice experiment, and it's subject to an experimental design. And we do rigorous development of the survey. The survey itself involves patient partners, and we make sure that the language is patient friendly, that you tell them a bit about RA, tell them what they need to know about the benefits of RA treatment, the risks of RA treatment, so that you can then go into the choice tasks. The modeling is, in this case, we use random parameter logit models, and we ran one model for the general public and a second model for first-degree relatives. In order to set up the experimental design, one of the first things you need to do is to identify the key benefits and key risks. And as you see here, I'm not going to go and read every one of them. There were 6 attributes. The first one was the chance to develop RA. So this is the effectiveness or the efficacy measure that we were telling people about. And if you were to take a treatment, hypothetically, you could lower your risk of getting RA could be lowered to 10%, or it can be as high as 40%. So how the treatment is taken could matter to people. And again, these are trade-offs, just as you heard in the first presentation. You make trade-offs between these attributes in a discrete choice experiment. So the risks here are mild side effects, a chance of serious infection because most treatments for RA suppress the immune system, and the chance for a serious side effect. And so you see, this is the experimental design underpinnings. And then from this, we design the survey, and there are different efficiency algorithms that you can do to make sure you cover the space, even if you only ask 10 or 15 questions per person. So the other thing that preference studies have is a vignette. And I'm not going to read this one to you fully, but it talks about, imagine you're at risk of RA, but there could be a medicine. And this is what's going to happen, and your doctor suggests a treatment. Which one might you prefer? And you could also pick nothing. So that's considered an opt-out. This is okay, it's easier to read for you than me. This is one choice task. So each person, subject to an experimental design, gets 15 choice tasks. And I think in this case, we also had 10 blocks. And so this is a block design. Each person gets 15 choice tasks, and you vary it. So the treatment A is on the first column. You can see that the chance of developing RA is reduced from 60% to 20%. So that's the first thing the patient might notice. Treatment B doesn't reduce it as much. It goes from 60% to 40%. So then you have to look and see, how is it given? And what are the serious chance of serious side effects? So as with any trade-off, the treatment A will give you more efficacy, but in this particular choice example, it will give you higher risks of some of the safety events. So, every benefit risk problem is these trade-offs. What are you willing to do to get more efficacy? in terms of the trade-off with the risks. I'm going to present results around our data quality metrics because we're talking about comparing these two samples and also the preferences themselves. There's a lot in the literature around figuring out how well people are paying attention to your survey. Are they speeding and just clicking to get to the gift card? Are they picking A every time? Or are they actually paying attention? So we do, during the survey and the warm-up questions, when we educate around RWE and the benefits and risks, we do ask a series of questions. And they're to test that you can understand, because we also look at literacy and numeracy measures. Do you understand it? Are you racing? And can you answer the questions that we've embedded, which is a measure of sorts for quality. We also ask people how easy or hard it was to understand and to complete the survey. There's a body of literature here with John Bridges and others in the preference space that when you get numbers below a certain threshold, something might have gone wrong with regard to how the patient was able to take the survey. The survey might have an issue. Preferences themselves come out of the model by looking at the model parameters. And then to get the maximum acceptable risks, which I'm going to spend a little bit of time on in the results, you actually do ratios of the parameters. There's a lot I'm not going to talk about with regard to, do you assume linearity or not? Do you do effects coding? There's a lot underpinning the model itself, but maximum acceptable risk allows us to know the maximum acceptable risk of a severe adverse event in exchange for a certain efficacy improvement. And so this is something we look at too, to make sure that people are interested in a treatment, but there might be some risks that are too high and they would not pick the treatment. And this involves the weights that come out of the model. So I put on some select results here. And the first thing is the time to complete the study. I mentioned you can speed through. Well, here, there's a difference in the time spent on the study. Now, there's the general public here is on the left. They took a median time of about 16.5 minutes to 29.8 minutes for the first degree relatives. So you can see there's a difference here. First degree relatives are taking more time. There are a lot of reasons that this could be, but we'll start with this observation. Comprehension questions, the general public did not perform as well as the first degree relatives. And finally, we ask how easy is it to understand and how easy is it to complete? And there were some differences here. Interestingly, first degree relatives report, they have fewer people reporting that it was easy or very easy to answer. Again, there are some reasons for this. I think there are also some differences in patient characteristics here, and in the paper I get into more detail. However, this does paint a picture that the first degree relatives are a bit closer to the problem and they're taking a bit more time. And because the risk for them could be very real and they have a first degree relative with RA, depending on what they notice in their first degree relative, they're really taking their time to answer the questions we've asked, and they might have more difficulty because of their proximity to the problem. And there's some things in the literature about this around psychological distance. I left this bullet, but I'm not going to read it in case people are downloading the slides. That's the punchline. So here's the model results. We look at the model and rescale to make the highest attribute size 10. So here you see that the most important attribute is the efficacy one, the chance of developing RA. And the least important one is the chance of mild side effects. So I have my two different samples and I say, okay, well, for the most important thing, my proxy, my general public did okay. And for the least important thing, they agreed. In the middle, it's generally similar. And then the red asterisk is there was definitely a difference in terms of how they ranked. The two populations ranked the mode of administration. There's some things about injections and things like that. Again, I'm not going to read these. They were for people who were not here. With regard to the maximum acceptable risk, just very briefly, there are differences in terms of the fact that the first degree relatives will will take a higher maximum acceptable risk on average than the general public. And this plays itself out by looking in particular, look at just mild side effects. They'll take an 80% chance of mild side effects versus 40. Some of them, these are not statistically significant. So my last slide is in the context of patient preferences, we're very quick sometimes to use the general public, and they might be reasonable if you just want to know the best or the worst attribute. However, when you start to get into maximum acceptable risks and the actual weights, there could be problematic issues. So a proxy sample like the general public that we tell them to pretend they have a disease needs to be carefully justified. Okay.
Just one question. Wanted to understand, when you looked at first degree relatives, did you do any further stratification to look at if they were actual caregivers of those, of patients with RA?
We did, and we are, and that will be a little bit in the discussion section of the paper.
Okay.
Clearly, the people that were closer to the caregiving and closer to the
more.
first degree relative have higher maximum acceptable risks.
Thank you.
And again, it relates to the psychological distance that I talked about.
Hi, Rania Mushanta from Health Canada. We do a lot of surveys in Canada, but I was just wondering, one of the major challenges is participation. How do you, any tips and tricks to enhance participation, especially from the public, if you have?
I don't know that I have the answer because it's something we debate all the time. We go to panels because they're convenient, but there are issues. I've recently had more success in the rare disease area when you go to the patient groups. The criticism when you go to publish is that, of course, you went to a disease group, they're different. But in terms of rare diseases, that's where you're going to be most successful at getting activated patients who want to participate. And we've had a lot of success with the patient groups themselves. And so we're going to be talking about sampling and how you find patients and how you get them to participate for a long time. I wish I had a better answer.
Thank you.
If I would be a first degree relative, I would ask myself how long the duration, how many years I need to take the prophylactic treatment. When can I stop it? That would worry me. Is there an age where I can stop it?
Well, yeah.
I know the answer, but I myself, this would be the problem.
Same for me. And I think we're out of time, but I'll say these studies did read out and it's not durable. You delay it, but when you stop, people at risk of RA get RA. And so when the studies that I've read out show that it's not as promising as we thought. And the minute you stop, the trajectory continues.
Thank you. And next speaker on the floor is Shahab Atabi from Utrecht University. Welcome.
Yes. Hello, everyone. My name is Shahab Atabi, Assistant Professor in Pharmacoepidemiology from the Division of Pharmacoepidemiology and Clinical Pharmacology at Utrecht University. I would be very happy today to present you my study on the impact of 2018 EU risk minimization measures and revised pregnancy prevention program on utilization and prescribing trends of valproates. Well, this was a large multi-center study with lovely co-authors from several European countries, including 6 different organizations. The manuscript of this project has been recently published in Drug Safety. We received funding for this project under a framework contract from European Medicines Agency. Otherwise, I have no conflict of interest to disclose. Valproic acid or valproates has 3 indications as epilepsy, bipolar disease, and for prophylaxis of migraine. But it is very well known for its teratogenicity and congenital malformations and neurodevelopmental disorders. Due to this teratogenicity, there were some risk minimization measures in place by European Medicines Agency in 2014 in terms of educational material for doctors, patients, DHBC communication, updating the package leaflets and SMPC. But a review by the Pharmacovigilance Risk Assessment Committee, PROC, of EMA revealed that the awareness of the teratogenic risks of valproates was low in 2018. The adherence to the educational materials was also low, and there were still pregnancies happening when exposed to valproate. So some strengthening of those measures happened, and ratified also by the European Commission in 2018 in terms of adjusted risk minimization measures and some pregnancy prevention program to really avoid the use of valproates in women of childbearing age, actually to contraindicate it for some indications, and if we cannot find any other alternative, to allow it for use only if the conditions of a PPP, pregnancy prevention program, can be met, like rigorous pregnancy tests, use of contraceptives before, during use, consultation with a specialist, and some filling in some acknowledgement forms, etc. So the aim of our study was to investigate the use of valproates in EU before and after the implementation of the 2018 risk minimization measures, and to see the effectiveness of the 2018 intervention using longitudinal data collected from 5 electronic healthcare databases from 4 EU countries and the UK. This was a huge project comprising different objectives, so utilization of the valproates itself, but also we went to check for a contraceptive coverage during and before the valproate use, the utilization patterns of alternative medications, and at the end, the concurrent pregnancies with valproate exposure. The protocol of this study is already registered in EU PASS, and it bears an NCEP seal. As I mentioned, we used data from 5 electronic healthcare databases in Denmark, Italy, Netherlands, Spain, and UK, with different characteristics. The design was a population-based cross-sectional time series study, which includes all females of childbearing age between the ages of 12 and 55 from the included databases between the beginning of 2010 until the end of 2020. Valproates and their alternative medications were the exposures of interest, and we estimated the incident use, prevalent use, discontinuation rates, duration of use, and switching from valproates to alternatives. The outcomes were the contraceptive use during valproate exposure and the concurrent pregnancies. We estimated the counts, some descriptive analysis, some counts, percentages, proportions of different outcome measures, but also we conducted time series, interrupted time series analysis in terms of segmented generalized least squares regression analysis to see any difference in the outcome measures before versus after the 2018 intervention in the data sources. We conducted rigorous quality checks to ensure that data is of, enough quality for the analysis, and we also conducted comprehensive benchmarking of the results. In order to get the data from different sources and to harmonize and standardize the heterogeneous data from different sources in the centers that I mentioned, we used the federated analysis approach by using all tables of the conception common data model. As you see, we get the, can I use the pointer? Yes. So here you see this is the raw data actually from different sources, and we do the process as ETL, extract, transform, load. We turn it into some common tables ready to analyze, and then to produce the results and visualization. And the common analysis script is open source and is available on GitHub. Let's take a look at the study results. At the end, we included almost 10 million females of childbearing age. Half of them were coming from BFOP, and but also from the other centers, as you see. Almost 0.5% or below 1% of the included patients, included individuals were using valproates during the study period. I think the only exception was Ars Toscana, when 2.6% of females were using valproates. The median follow-up time in most centers were close to the whole study period, as 10 or 11 years. The exception was CPRD here, it was with 4.5 years. And the mean age of at index date were all above 30 years of age. For the sake of time, well, we produced a lot of results, a lot of plots with ITS analysis. I only show you the prevalent use of valproates among females of childbearing age. This is the overall trend in Denmark. Due to some restrictions with the Danish data, we didn't have enough time points after the intervention to conduct an ITS analysis. And this is the overall trend across the study period from the beginning of 2010 until the end of 2018. The dashed line shows the intervention time sometime in summer 2018. This is the results for other centers. So in the left, you see the main study period from 2010 until 2020. This is for Ars Toscana in Italy. As you see, there was a significant decline in trend in valproate use after the intervention. On the right side, you see our sensitivity analysis for the period between 2015 until February 2020, which actually excludes the time before 2015, which was the time of the first risk minimization measures in Europe in 2014, but also excluding the COVID pandemic time after the February of 2020 in Europe to get away from any effect from the pandemic time. We still had a significant decline in Ars Toscana. In Pharmo Netherlands, we had a marginal decline in trend in valproate use in the main analysis and non-significant changes in the sensitivity analysis. This is the Spanish results from BFOP. We still had a significant decline in prevalent use after the intervention, which remained the same. Also added to the significant decline in level in the sensitivity analysis. And in CPRD, we also had a significant decline after the intervention in trend of valproate use, but non-significant changes in the sensitivity analysis. This is the quantification of the impact of the 2018 risk minimization measures. So I try to summarize everything here in terms of the mean difference post-intervention compared to the counterfactual values had the intervention not occurred. And in parentheses, you also see the percentages. So like here, you see we had 7.7% decrease of prevalent use, significant decrease in Ars Toscana, also significant decreases in BFOP and CPRD. Well, for the other objectives, we didn't find many positive associations. It was only for Pharmo, I think, the contraceptive coverage, 12% increase in contraceptive coverage after the intervention. And this is the last slide of my results. We observed substantial number of concurrent pregnancies during valproate exposure, almost in all centers. But the good news was that, well, the rates were declining after the intervention in all centers, apart from CPRD, where the rate was actually increased. These are the strengths and limitations of, I would go very quickly through them. So very comprehensive study and with generalizable to the European setting, 10 million people involved, used some sort of federated analysis, common data model, as I said, to harmonize the heterogeneous data into something that we can easily run the analysis on. And we also conducted rigorous quality checks. The limitations were, well, for some of the objectives, we could not capture the information because they were over the counter, like contraceptives, but also pregnancy tests, as was one of the initial objectives that we, in the terms of the project, we actually put it away. And we also had some limitations in the Danish database, as I mentioned, and some weird results in CPRD Gold, which we could not fully understand. And also limitations because of COVID pandemic, etc. So in conclusion, there was a small impact of the 2018 risk minimization measures on valproate use and prescribing trends in European countries. The substantial number of concurrent pregnancies with valproate exposure warrants a careful monitoring of implementation of the existing PPP for clinical practice in Europe to see if we need to do something or to add something to it in the future. Thank you very much for your attention.
Thank you, Shahab. And one question.
Yes, please. Thank you very much, Priya Vai from EMA, working on valproate as well. I have two questions. One is about the decline. The users you currently see, do you think this is compatible, or do you have data in terms of indication with the very small indication which is still applicable to women of childbearing potential, or do you think there is still an over prescribing, so to say, or off-label use, if I can call it like this? And how have you counted the pregnancies? Are all those pregnancies which have begun but possibly could have been also terminated, or have you only counted pregnancy which went through? Thank you.
Yeah, thanks for the excellent questions. Regarding the indications, well, those were definitely one of our parameters to include and to see for them in the beginning. But unfortunately, I think in all the centers, apart from all the harmonization, as I said, and quality checks, etc., I think the indication for valproate was like around 90% or more than 90% unknown. So that might be off-label use, I'm not sure, or that might be the problem in recording of data in different databases. So that was about indications. And for pregnancy, so I didn't show it here because, as I said, this project was huge. We use an algorithm and a revised version of a previously validated algorithm to detect pregnancies in centers based on very different information from imaging, or some other parameters during the fetal life. Yeah, very different parameters we used to actually estimate the start date of pregnancy, but also end date of pregnancy. And we also marked them with different colors. So when we were really certain that, well, there was a pregnancy there, we called it as green, otherwise it was red or yellow, and we know that we're really running low in time. So.
Very much. Excellent question. I know you have more questions, and please follow up after the session as well with more questions. Now we're going to move on with Nicole Smolowski. She is a graduate PhD student at the University of Florida.
All right, so we're going to look now at the US side of valproic acid, so very similar to what we just heard. This is a project in my pregnancy team at UF. I do disclose some stock via inheritance. In terms of background, as we just heard, valproic acid was originally approved for epilepsy, and then its use expanded. So now it includes migraine prophylaxis as well as bipolar disorder. There's a lot of evidence to show that it's a teratogen. We see neural tube defects, we see neurodevelopmental defects. As we just saw, a lot of regulatory agencies across the world have put in place new warnings, new risk minimization programs to kind of reduce this exposure to pregnancy. So one thing our group has done in women of childbearing age, or sorry, persons of childbearing age in the US is look at valproic acid use across the timeline. So here I'm showing from 2005 to 2020. The vertical lines show the different label changes and drug safety communications that the FDA put out. And you can see that both in males and females, there's a decreasing trend in valproic acid use. It's slightly sharper in the females, but the males seem to also be decreasing. So then what we wanted to do was look at the indications. So this is specifically in females of childbearing age. We looked at the different indications and how that trend is looking across the study period. So we see that the majority of patients are using it for mood disorders. Unknown is our category where we're not really sure what they're using it for. It likely is that off-label use that we know is existing. And we see not quite as sharp of a decrease in the patients with epilepsy and then the migraine disorder patients. So because we saw this decrease based on indication in kind of the general women of childbearing age cohort, we wanted to see, is there also a difference in whether or not pregnancy occurs in these patients? So the aim of our study here is to determine the rate of pregnancy onset during valproic acid use, as well as the extent of contraception use within that population. So we used Market Scan Commercial Claims Database. So this is private insurance in the US from 2005 to 2020. We're looking at female patients that are aged 12 to 55 that had continuous health plan enrollment for 180 days before treatment through 270 days after the end of treatment. That is specifically so we can find pregnancy, and we have a full 9 months to see the pregnancy end. We looked at patients that had at least one outpatient dispensing for valproic acid during our study period. And then in that 180 days look back prior to the dispensing of valproic acid, that's where we looked for our indication. For our pregnancy identification, we used our group's validated pregnancy algorithm to figure out conception date, pregnancy endpoint, and then the pregnancy outcome. And then for contraception, our team also has a contraception algorithm. So we looked for at least 1 day overlap between the valproic acid and a found contraception method. The figure here is to kind of help show how we figured out pregnancy and treatment episodes. So we made treatment episodes of valproic acid where we allowed gaps between fills of 7 days. And then we simply were looking for a conception date that overlapped with that treatment episode. For our incidence rates, we calculated annual pregnancy incidence rates, as well as contraception use prevalence. For the pregnancy incidence rates, we did crude as well as adjusted. And then we also saw that there was an age distribution, or we assumed there would be, so we did direct age standardization using census data so that we can kind of account for the age differences based on indication. So some basic demographic information, we identified 268,000 valproic acid treatment episodes. The vast majority of them were mood disorders, as we expected from our previous work. The average age was 37. But we saw the epilepsy patients were a little bit younger. Their average age was 33, whereas the mood disorders were a little bit older at 39. In terms of the region of where the patient was from, the majority was in the South. That is kind of part of our data. Most of Market Scan is patients that live in the South. In terms of the average treatment episode length, so this is just on average, how long were the treatment episodes that we constructed? The overall was 92. We did see that the epilepsy patients had slightly longer with 111. So looking at our incidence of pregnancy. So this is our graph. You can see it's pretty stable across the study period. There's some kind of up and downs, but we saw maybe a tiny increase in 2005, it was 0.9 per 100 person years, and it slightly increased to 1.2. To look overall at our actual rates across the entire study period, we identified 733 pregnancies across the study period. The majority of those came from the mood disorders. There was 379. I want to put everyone's attention to the last column just so that we can look at the overall. So this is the adjusted incidence ratio. These are adjusted for both calendar year as well as age of the patient. And here's where we can actually compare the different indications. So we used epilepsy as our reference group, and you can see for both migraine and mood disorders, our ratio is greater than 2. So these patients are significantly more likely to get pregnant than those epilepsy patients. That unknown category is also a little bit higher at 1.4. In terms of pregnancy outcomes, we had 61% were live birth, which is actually lower than we were expecting. And if you look at it, the epilepsy patients are even lower at 55%. So this is something that our team kind of like pointed out of it's less than we would expect. So it's something we kind of want to look into of why this is happening. The other thing that was interesting was the average pregnancy days exposed. So this is essentially how long once they're pregnant do they stay on valproic acid. And you can see that for epilepsy, they do have slightly longer average time that they're staying on the drug. So this may be because their physician has talked to them and they want to stay on the drug longer. In terms of contraception, we found very limited contraception use among our users. So I'm showing the patients that there was no identified contraception. So 85% of our patients, there's no identifiable contraception. That number kind of varies by indication slightly. In terms of the trend, so you can see across time, we do see a slight increase across the study period. So it does look like there might be a little bit more adoption of contraception across the study period. As expected, the most of our patients were using oral contraception, followed by injectables and IUD. So in conclusion, the pregnancy incidence during treatment with valproic acid is varying based on the different clinical indications, and what we're calling the risk mitigation behavior of contraception is quite uncommon in these patients. So kind of our overall recommendation and conclusion is that there might need to be more warnings and patient counseling, specifically in patients with migraine, headache, and mood disorders, because that's where we're seeing the highest risk for prenatal exposure. So there might need to be more appropriate prescribing and patient counseling, and maybe focusing risk mitigation on those patients. Thank you. I will take any questions anyone has.
Thank you. It's very, very short, and you might have said it. Is the contraceptive reimbursed in your system, or the women?
Yeah, so this is anyone that had a prescription or a clinic visit.
But is it automatically reimbursed for somebody who has epilepsy?
So contraception is covered under our Affordable Care Act for everyone. So as long as they have private insurance, it's covered.
Any other questions? Very good. No other questions? Excellent presentation. I have one. In terms of decision-making,
Mhm.
what are the key limitations that need to be considered before making decisions? I know you mentioned some recommendations, but any additional limitations that you need to consider that would be key in decision-making?
I mean, the biggest thing that everyone always brings up is barrier to access. So if you put something into place, are you limiting people's ability to actually get access to the drug? I would say I'm less concerned about that because there's already a decrease over time in the use of it. Because I think there's a lot of alternatives that are available to patients that they're kind of being switched to, but the patients that are staying on it are not. And those are the patients that are now at risk.
Very good. Any other questions?
Yes, I also had an additional question. So you saw this difference in pregnancy rate between the different indications. Do you have an idea of why that might be?
Yeah, so one of the things that our team potentially thinks is a lot of the evidence for why valproic is a teratogen is specifically in epilepsy patients. So it's possible that it's only the neurologists that are truly aware that it's a teratogen. So the primary care providers that are prescribing patients for migraine or mood disorders aren't necessarily aware that it's a teratogen, and that could potentially explain why we're seeing less pregnancies in the epilepsy patients.
Hi, Alicia Gilston from RTI. That was actually going to be my question, and maybe just expanding or reminding in case I missed it. What was the latest communication, and was it a Dear Healthcare Professional letter, or is there more specific communication, and is it targeted to a specific?
Yeah, the most
provider?
recent for the FDA was just a label change to specifically add the risk of the neurodevelopmental, because that is more recent that we're finding out that that's part of the teratogen.
Good afternoon, everyone. I hope everyone is the food coma really started to hit me. So please stay awake with me. So in this presentation, I'll continue the topic from the previous two presentations, focusing on evaluating the risk evaluation and the mitigation strategies for the prevention of fetal exposure to phentermine topiramate and using two large US nationwide cohorts. So I have no conflicts of interest to disclose. So first, to provide some background about phentermine and the topiramate combination treatment. So phentermine has been used for short-term treatment for obesity. And in 2012, US Food and Drug Administration approved the combination of phentermine and topiramate to treat obesity as well. So topiramate has been shown to cause oral clefts, and the FDA, while they are approving the combination treatment, due to this concern, they required a risk evaluation and a mitigation strategy to prevent fetal exposure because of the concern of oral clefts due to the topiramate exposure. So pregnancy tests were required for the patients to have before they start the combination treatment, and they are also required to have monthly pregnancy tests. In addition, birth control methods are also required while the patients are on treatment. But we are not really sure how effective the RAMS is in preventing pregnancy occurrence among the patients who are using the combination treatment. So the objective of this study is to evaluate the effectiveness of phentermine topiramate RAMS for prevention of pregnancy occurrence. So we will use quite a similar design compared to the previous two studies as well. So in terms of the database, we use the both Medicaid data, which is a publicly insured population in the US. And we also have the market scan research database, which was just in Nicole's presentation, focused on the privately insured population. So we included a woman aged 14 to 45 years who initiated the treatment. So for the exposure group, we included the ones who initiated phentermine plus topiramate combination therapy. And then for the control, included patients with phentermine treatment. So we required 90 days, at least 90 days before the index date, which is the initiation date, to require the continuous insurance enrollment to capture the information. And we further require the patients do not have sterilization procedures, diagnosis of menopause and premature menopause during baseline, because those patients will have a different probability of becoming pregnant. So using the pharmacy claims data, we try to create treatment episodes for both the exposed and the control group. So we captured first all the dispensing of the both treatments. And then for any consecutive dispensing, we check the gap between the end of the day supply of the first dispensing and the field date of the second dispensing. So if the day supply, the gap between the two dispensing is less than or equal to 30 days, we consider them under the same treatment episode. If it's more than 30 days, then they will be starting as a new treatment episode in our data set. So then based on this, we assign the index date and also the end of treatment episode to evaluate when the patients are on the treatment and when we can further look into whether they become pregnant afterwards. So in terms of the outcome, so we measured the occurrence of pregnancy. We utilized the hierarchical pregnancy algorithm to determine different pregnancy outcomes, because in the database, as we discussed in the previous presentation, so basically we need the pregnancy codes first to determine whether the pregnancy occurred in the database, either it's a live birth or non-live birth. And then after that, based on the data of the occurrence, we look back to determine the start of pregnancy. Because what we really care here is when they are on treatment, do they have pregnancy occur? So then based on the gestational age algorithm, we can estimate the start of pregnancy, and the patients were followed from the index date until the earliest of the following days. So either it's the end of continuous enrollment or study period. We also added 30 days at the end of the day supply of the last dispensing, basically the end of the treatment episode, just to account for potentially the patients still have some leftover treatment. And we also follow the patient up until either they have a pregnancy actually occurred based on the start date. And for the phentermine group, which is a control group, when they switch from phentermine to phentermine plus topiramate, we then stop the patient's follow-up the day before the field date of phentermine and topiramate combination field date. So for the main analysis, we estimated both the crude and the adjusted incidence of pregnancy in both groups. And then we further compared the two groups, basically the pregnancy occurrence between the two groups, and estimated hazard ratios using the Cox proportional hazards model. We also conducted a post hoc secondary analysis where we stratified the main analysis by race and ethnicity groups in Medicaid. We were not able to do so in market scan data because race and ethnicity information is not available in market scan data. And that's coming from basically some observation and discussion regarding the RAMS program have a different effect among different racial and ethnicity groups. So moving on to results. So first, in both the Medicaid and market scan databases, we observed that compared with patients on phentermine topiramate are more likely to be Hispanic patients, initiating the treatment in more recent years because the combination treatment is approved more recently, although we use a period after their approval to conduct the analysis. And they reside in the West region and also use more likely to use contraceptives during baseline. So we first moving on to the results of the main analysis, where we looked into the incidence of pregnancy among women who were treated with either phentermine topiramate versus phentermine. So if we first focus on the Medicaid population. So here I'm presenting both, first the three rows are focused on more like the numbers of episodes and the number of pregnancy during the follow-up time. And then if we look into the crude incidence rate of the pregnancies occurrence in the fourth row, we can see that we have about a similar incidence rate in both the phentermine topiramate group versus the phentermine group, and then which leads to a crude and adjusted hazard ratio around 1. So basically here, we don't really see a strong difference. If we interpret this, the occurrence of pregnancy between the two groups indicating the effectiveness of the program, then we don't really see a strong difference between the two groups. But when we look into the market scan data, which is a privately insured population, so here we have a little bit more sample size in both treatment groups. This is a different population, more likely they are receiving treatment for obesity and also more recent years of data as well. So here to begin with, within the phentermine group, we actually observed a lower pregnancy rate to begin with. This is also potentially due to the two population is quite different, the privately insured and versus the publicly insured population. And then if we look into the phentermine topiramate group, the crude incidence rate is actually 0.5 per 10,000 person days, so which is even lower compared to all the other groups we observe in both databases. So if we compare the two groups within the market scan database, we observed a crude and adjusted hazard ratio around 0.54 for the crude, and the adjustment did not really make a lot of difference. Here we adjusted for age and calendar years. So if we look at the results in market scan data, then we will see actually there is an effect of the RAMS program to prevent pregnancy within the phentermine topiramate group when compared to the phentermine group. So we also were curious about, among the patients who become pregnant, what kind of pregnancy outcomes we actually are observing using the two database. So we looked into both occurrence of live births and also non-live births, including termination and spontaneous abortion. So in the across both Medicaid and market scan databases and across the two treatment groups, we observe a quite similar distribution, except we observed in phentermine group, the termination percentage of termination is a little bit lower in the market scan database. But most likely this is due to the fact like we have a limited number of patients that actually become pregnant to start with. So basically we have a quite small denominator here, and the estimate may not be quite accurate or stable across the different the two groups within a single database. So after we observe the difference between the publicly versus the privately insured population, one thing we start to think what can potentially address what we observe here between the two populations, why there is a differential effect of the RAMS program for phentermine topiramate. So we then looked into using Medicaid data, because here we have racial and ethnicity groups information. So we further stratified the analysis based on the race and ethnicity groups. So I'm listing here in the table the first main columns are the overall estimates, which I have already reported on the previous slides. And then first, let's take a look at among the Black and the Hispanic patients. So for the Black and the Hispanic patients, what we observe in Medicaid when we stratify by racial ethnicity groups, the incidence of pregnancy is close to the overall estimates, slightly higher. And then when we look at the crude hazard ratio, we also observe a similar estimates. We don't really observe a strong effect of the RAMS in terms of preventing pregnancy among those two groups. But when we take a closer look into the white population, so here we observed within the phentermine group. So we have the pregnancy rate is 1.85 compared to the other two groups. We already observe a slightly lower pregnancy rate. And then in the exposed group, which is a phentermine topiramate group, we observed an even lower rate of pregnancy. So then leading to a crude hazard ratio of 0.67 based on the point estimate, although we have limited sample size to further, so leading to a relatively wide confidence interval here. So what we observed here is potentially the differential effect we observed between the privately versus the publicly insured population is really potentially related with the race ethnicity groups. And then the point estimate within the white population is actually now becoming closer to what we observe in the market scan data. So in conclusion, in the commercial insured patients, we observed an effect of the RAMS program in terms of prevention of pregnancy occurrence among the phentermine topiramate patients. However, we did not really observe such an effect among the publicly insured patients. So after we looked further into by racial ethnicity groups, we observed that compared to Black and Hispanic patients, white patients had a lower incidence of pregnancy while being on phentermine topiramate treatment. So we think that potentially this is an area to look further into this because, we do care about, how can we best implement the RAMS program to prevent pregnancy occurrence among those patients who are exposed to those medications, potentially carries a risk for birth defects. So future research is required to examine the apparent differential pregnancy rates among the two publicly versus commercially insured population, plus potentially looking into subgroups as well. So I'd like to acknowledge my team and all the co-authors who contribute substantially to the project, and I'm happy to address any questions. Thank you very much.
Thanks for the nice presentation. As we obviously saw in the results, so there was a huge discrepancy between the two data sources that you used. Do you know what was the reason? It is because of the different characteristics of the data that we had from these two data sources, or different study population they covered, or anything else?
So are you referring to the different effects of the program, or are you referring to a different rate of pregnancy?
Different rates and the different effects that you showed us from the market scan, I guess, and the other data sources.
Yes. Yeah. So basically, let me explain a little bit more. So for the Medicaid population, typically it's low income population which receive Medicaid insurance, the publicly insurance in the US. So it does have a different composition of racial ethnicity groups and potentially also social demographic, differences between the two populations. And versus the market scan population, they are actually commercially insured. So they tends to be, they basically receive the insurance coverage through their employment. So it's a slightly different, the two groups are quite different in terms of that. So potentially our research group did, once we observed the effect, we were like, oh, how could we really, figure out what's exactly going differently between the two groups to explain the results. And the one thing occurred to us, it could be from the patient level, patient side, or versus the care they are receiving. Because, the RAMS program covers both. So they have a patient education plus education to the clinicians, which should implement the RAMS program. Yeah.
You partially answered my question, Rachel DeSanto-Stefano from Janssen. Whenever I find race findings, my question is it because it's Medicaid, do you think it's the care setting and the knowledge of the doctor and sort of the quality of care? And I almost hate to ask that, but I believe that's one potential explanation.
Yeah, one idea from one of our co-authors raised actually started to make me think more about it. So because we do observe a higher pregnancy rate in Medicaid, potentially, those programs, when you have a higher baseline rate, it could be unintended pregnancy, which is a large proportion, among the US population. Even if you add a RAMS program, it's potentially harder to bring down the rate further. Versus a commercial population, you have a rate which is lower, and then it's relatively easier to bring it down. But still, even given that, I think the other side is potentially we should think a little bit more about how these RAMS programs are being implemented. How does it impact the behavior of the patients? And also how does a clinician actually ensure? Do they, but what we reveal here in the, the secondary analysis, potentially basically based on different racial ethnicity group, based on the initial results, it seems like they are receiving different kind of care, or they are receiving different type of information, and leading to how they couldn't implement the RAMS, effectively in the same way, basically.
Just, you may have said this again, but can you remind us what the RAMS program involves?
Yes, so the RAMS programs for this medication specifically, it's not super strict compared to, let's say, isotretinoin. It requires a pregnancy test before the initiation of the treatment, and then monthly pregnancy test while they are on the treatment. in terms of contraceptions, so they require either a highly effective one, like a method, they only require one method, but let's say if it's through like condom or other like less effective methods, they require two combination. But we also have another project ongoing, so we actually observed that sometimes it's not actually adherent to the actual RAMS program, although it's designed in a way to all targeted towards prevention of pregnancy.
Yeah, that could have a major impact on the care setting if they're not following the program.
Yes, exactly. Yeah. Yeah, thank you.
Thank you. Thank you all for being here. So another risk minimization study. My name is Marius. I'm a doctor, a medical doctor and researcher at the Copenhagen Phase 4 unit in the capital region of Denmark. Today I'm going to talk about risk minimization activities in baricitinib treatment of rheumatoid arthritis, or RA, or the more official title, Descriptive Observational Cohort Study to Assess Effectiveness of Risk Minimization Activities in Baricitinib Incident Users Treated for Rheumatoid Arthritis in Nordic Countries. This was requested by the EMA, this study, related to the approval of the drug in Europe, and it's fully sponsored by Eli Lilly, which is the manufacturer of baricitinib. So, quick background, baricitinib is an oral JAK1 and 2 inhibitor used to treat moderate to severe rheumatoid arthritis, among other autoimmune diseases. It's superior to, for example, the biological DMARDs because it's a pill, so it's easier to take as a patient. It's in the category of targeted synthetic DMARDs, so disease-modulating anti-rheumatic drugs. It was approved in Europe in 2017, and this study aims to monitor the risk minimization activities or to monitor the adherence to the recommended risk minimization activities. So specifically for baricitinib, there are 3 general risk minimization activities that healthcare professionals should be aware of. First is to rule out active tuberculosis, viral hepatitis. Common side effect of these types of drugs are infections, and if a patient already has active tuberculosis or viral hepatitis, the disease tends to flare up. It's also not recommended to use the drug during pregnancy since there isn't any data of the safety of the drug. Yes, and the last one is the assessment of lipid parameters 12 weeks after initiation of the treatment, because baricitinib has a tendency to increase the lipid levels, the blood lipid levels. So this study had a population from the 4 different Nordic countries, Denmark, Norway, Sweden, and Finland. We had most patients from Sweden, and I think it totaled just under 40 or 4,000 patients total. So these were all incident users because it wasn't approved before the time. And the patients were included from different time periods because the data from the registries from different countries came in at different times. So that's just important too. We can see here that the patients in Denmark were followed longer than, for example, the patients in Sweden. These were all patients from the national patient prescription registries and the registries for inpatient, outpatient visits. So we classified patients as having the treatment exposure if they were incident users. Of course, they had a ICD-10 code, procedure code related to baricitinib, or they had a registration in the prescription registries. And if 2 different registrations were present and with a maximum of 180 days between them, we classified it as continued treatment. We followed these patients until treatment stop, until death, migration, or end of study. The treatment stop was 180 days after the last prescription, or if they started in some other DMARD. Yes. So we looked at these 3 different outcomes. So first up, infections. We looked at visits with ICD-10 codes related to tuberculosis and viral hepatitis. For pregnancies, we looked at an overlap between baricitinib treatment and pregnancy. Either the women were pregnant when they were started their treatment, or they became pregnant during their treatment. We used the registries with births, stillbirths, and abortions from the countries. And we also looked at change in lipid level. Here we looked at visits with hyperlipidemia diagnosis attached, or change in statin prescription. These were the 3 outcomes related to the 3 risk minimization activities. So first up, the infection visits. We can see here that very few patients had any tuberculosis diagnosis visit 5 years prior, and also maybe this was 0. This is a result of how the registries work. We can't report the data if it's less than 3. Viral hepatitis, we had a bit more patients with viral hepatitis diagnoses visits 5 years prior, but it was also quite low during the treatment. As for pregnancies, we had a subpopulation of 815 incident users who were women aged 18 to 50, and with a median age of, or mean age of just under 40 years old. Among these women, there were 8 women with an overlapping pregnancy. They were a bit younger than the other subpopulation of women aged 18 to 50. It resulted in 4 live births. So 0.5% of this subpopulation of women. For change in statin treatment, we used a surrogate measure of, because we want to look at whether the patient was screened for their lipid levels 12 weeks after. At that time, we didn't have the blood sample registries as we have now, so we had to use some other surrogate measure. And we looked at initiation of statin treatment after starting baricitinib treatment, escalation of statin treatment, reduction of statin treatment, or change to a different drug. So we saw that 9%, a bit over 9% had a change in their statin treatment, was primarily initiation of statin treatment or the reduction of statin treatment after they have started baricitinib treatment. As for infections, we found very few dyslipidemia visits during treatment with baricitinib. Yes. So we concluded in the report to EMA that the risk minimization or the advised risk minimization activities are well followed in the Nordic countries, at least. Regarding infections, very few patients had any visits with the related infections. Pregnancies, it's a bit harder to conclude anything from the very few pregnancies and very few births. But we can't be 100% sure that there's a true overlap between treatment and pregnancy, because if a woman has some medication and she becomes pregnant, she could just stop taking the medication. That we wouldn't be able to see that in the registries. So it might even be fewer true overlapping patients with the treatment. As for lipid levels, the surrogate measures were quite low, so I'm not sure it's proper to draw any conclusions on that. And it should also be stated that this is an ongoing study, and the next step is to do a adverse effect study, which will come out in the coming years. Yes. All right. Thank you for listening.
Thanks for the nice presentation. I was actually wondering, so that was the thing that we also faced and discussed a lot during the Valproate project that we were doing with many different colleagues. So yeah, from the philosophical perspective, what is the aim? So should we have 0 concurrent pregnancies? I'm not sure about the teratogenicity of baricitinib, so how are the rates? Or so in the case of valproates, it's really crazy, but or oral retinoids also. So what is the, what is the target? So you said that, okay, we have a tick.
Yeah.
We were following the, the risk minimization measures, but up to what extent we should really follow that?
Yeah, that's a good question. I think it's important to state that, at least in Denmark, it's not contraindicated to prescribe baricitinib to pregnant women. If they are very sick and that's the best treatment, then it can be the right decision to do. And so a 0 would be good, but it's not necessary that we would consider that a success. And also because the way, as I mentioned before, the way the registries work, it's very hard to see if there is a true overlap or the patient has just stopped taking the medication when they're pregnant. It's different for valproate because there's a shown effect or shown teratogen effect of the drug. We don't know that yet with baricitinib. So, yes.
So just to add on, hi Marius, I'm Kristen Myers. I was going to introduce myself after, from Eli Lilly. And that exact question came up a lot when we were interpreting the results. And so that is the idea, is that if it is a risk minimization activity, you shouldn't be seen. So it would have been, bad if you saw a big bolus of pregnancies, for example, but there wasn't a comparator to kind of benchmark it against. And it was more of a judgment call that we made and argued about with the limitations known about how we are measuring this, and then also letting, the regulators decide as well. And I think that it was considered a low number, especially considering that we were pretty conservative in a broad way of defining possible pregnancy overlap with baricitinib.
Yeah.
But yeah, very good question, and we struggled with that as well. So,
yeah. And the EMA was also accepting the conclusion as well with the pregnancies. Yes. Any other questions?
Maybe in the light of the previous questions, I think for the JAK inhibitors, there's recently been sent out yet another risk minimization measure, again, a warning around, well, an iteration of some of the warnings here, and I think then some additional ones. So in that light, what effect could we expect of a additional, yet again, letter sent out warning about infections and VE and so on?
Sorry, what effect we would have from what?
Do you think we would be able to see any effect now that you've already shown these quite small occurrences of both infections and so on, sending out another letter for JAK inhibitors and less effect?
That's a good question. I think the results here speak for that it's good enough as it is, so maybe we shouldn't do more against it, spend more time on it, but yeah, I think it's as low as it gets, probably, at least for infections, as you mentioned. Adverse effects and other infections which can occur as adverse effects, that would be the next thing to study with relation to this baricitinib. So I think here we can improve, probably. Let's see when the data comes. Thank you.